Non-Mendelian genetics. Table of contents. Non-Mendelian inheritance, mitochondrial inheritance, inherited disorders, pedigree chart, environmental impact on phenotypes. Non-Mendelian inheritance. The relationship between genotype and phenotype is rarely as simple as in the pea plant characteristics Mendel studied. Many heritable characteristics are not determined by only one gene with two alleles. However, the basic principles of segregation and independent assortment still apply, even to more complex patterns of inheritance. Inheritance of characteristics from a single gene may deviate from simple Mendelian patterns when the gene has alleles that are not completely dominant or recessive, has more than two alleles, is subject to codominance, produces multiple phenotypes, is linked to another gene, is on a sex chromosome, and is in mitochondrial or chloroplast DNA. Complete versus incomplete dominance. What we've seen until now when studying Mendel's work is complete dominance. This occurs when the phenotypes of the heterozygote and the dominant homozygote are identical. In incomplete dominance, the phenotype of F1 hybrids is somewhere between the phenotypes of the two parental varieties. So if my parent generation is a red rose crossed with a white rose, I end with a pink rose. This pink rose results from the heterozygote having less red pigment than the red homozygote. This is not blending. If blending occurred, you would never be able to retrieve the red and white traits from the pink hybrids. This is not the case for incomplete dominance. It is possible to obtain red or white offspring after crossing two pink hybrids. In codominance, two dominant alleles affect the phenotype in separate, distinguishable ways. The ABO gene in humans has three alleles that exist at the same locus, or specific physical location of a gene or DNA sequence on a chromosome. The A and B alleles are codominant, and the O allele is recessive. Any one individual will have only two of them, one from the father and one from the mother. These three alleles in different combinations produce four phenotypes: O, A, B, and AB. Human blood types. There are different notations for the genotypes. When there is more than one dominant trait, the variations are often written as a superscript of another letter. In this case, capital I for dominant, lowercase I for recessive. Since A and B are codominant, the alleles are, we have the capital I for the dominant in A or dominant in B. There is no superscript for the recessive, lowercase I, as there's only one recessive allele, O. So let's look at these blood types. We have A, from the previous slide, A, B, AB, or O. Those are our phenotypes. The genotype is dominant for both, or dominant A and recessive O. So AA or AO. The alleles would be IA or IA dominant, and then the recessive lowercase I. It's the same for B. Okay. Now, if I'm AB, the genotype is AB, and I'm dominant in both A and B. Or if I'm O, then I'm recessive for both. Example 1, human blood types. Use a Punnett square to show the genotypic ratio and the predicted phenotypes of a child whose father has AB blood type and whose mother has AO. For simplicity, you can use just the letters A, B, and AO without the full written genotypes IA, IB crossed with IA and then the recessive lowercase I for O. Let's take a look on the next slide at the Punnett square. Okay, so let's take a look at our Punnett square. We have the AB on the left, contributed from the father, and the A or O on the top, contributed by the mother. So the resulting genotypes are AA, AO, AB, and BO. So all four of these genotypes are different. Okay, this is dominant for both A. This is dominant A, recessive O. Co-dominant for both A and B. Dominant B, recessive O. So our genotypic ratio is 1 to 1 to 1 to 1. But our phenotypes, two of these will have type A blood. Okay, so that's 50%. Right? Type B is just this one right here that's dominant in B and then recessive. So that's B. And then the one that's co-dominant, both AB, will result in type AB blood type. So 50% type A, 25% B, and 25% AB. Example 2. Using a Punnett square, show how the genotypic ratio and the predicted phenotypes of a child whose father has AO and whose mother has OO. So try to do this on your own before I show the next slide. So the father is AO and the mother is OO. Try to fill in this Punnett square and see what you get for your different genotypes and phenotypes. Let's go over it on the next slide. All right, so if the father is AO and the mother is OO, what blood types could result? So we could have AO, OO, OO. So we have two genotypes here that occur in a 1-to-1 ratio, AO to OO. What about the phenotypes? Well, those that are dominant in A are type A, that's 50% of them. And the one that has two recessive alleles is type O. So 50% type A and 50% type O. Chickens are another example of codominance. When black-feathered and white-feathered chickens are crossed, so we have dominant for white feathers, crossed with dominant for black feathers. When we cross those, we have a dominant allele for black feathers, a dominant allele for white feathers, and the resulting offspring are not black, white, or gray feathered chickens, but a chicken with both black and white feathers all over. So dominant black feather, dominant white feather results in a black and white chicken. Another example of codominance between black and white. So, what is the relationship between dominant and recessive alleles? A dominant allele does not subdue a recessive allele. Alleles don't interact. Alleles are simply variations in a gene's nucleotide sequence. For any characteristic, the dominance or recessiveness in relationships of alleles depends on the level at which we examine the phenotype. Pleiotropy. Pleiotropy is a situation in which a single gene may affect many phenotypic characteristics. Pleio meaning more than one, tropy meaning affecting or pertaining to. So affecting or pertaining to more than one phenotypic characteristic. An example of pleiotropy in humans is sickle cell disease. Sickle cell disease, or SCD, is a disease characterized by many diverse symptoms, such as abnormal red blood cells leading to physical weakness and anemia, clumping of cells and clogging of small blood vessels leading to pain and fever, possible brain damage, and possible heart damage, and accumulation in the spleen of abnormally shaped red blood cells leading to spleen damage. You can see the shape of a normal red blood cell here and the shape of a sickle-celled red blood cell here. All of the possible phenotypic effects of sickle cell disease are caused by the presence of a single allele when it is present on both homologous chromosomes. The direct effect of sickle cell allele is to make red blood cells produce abnormal hemoglobin molecules, which tend to link together and crystallize. This, in turn, leads to abnormally shaped red blood cells, which take on a crescent shape. Sickle cell trait, SCT. People who have inherited the sickle cell disease from one but not both parents have sickle cell trait. They usually don't have any of the sickle cell disease symptoms and lead a normal life. In rare cases, they may experience pain in high pressure areas, low oxygen levels, dehydration, and high altitudes. Athletes with SCT are more likely to have dehydration and muscle breakdown. Their genotype is heterozygous. However, there are benefits to having SCT. SCT people survive malaria infections better than homozygous normal, those who don't have the sickle cell gene, and can be the healthiest individuals in some parts of the world. This principle is called heterozygote advantage. SCD and SCT inheritance. So if there are two parents heterozygous for the sickle cell gene with SCT, so they carry the sickle cell traits, their offspring will have one homozygous normal, two that are heterozygous and carry the sickle cell traits, and one homozygous for sickle cell disease. Polygenic inheritance. Another variation from Mendelian genetics is that a single characteristic may be influenced by more than one gene. Mendel studied characteristics that were classified as either or, such as purple or white flowers. Mendel's pea plants were either tall or short. There was no in between. Each characteristic depended on only one gene. It does not always work that way in genetics. Many characteristics vary over a large scale in a population and are determined by many genes. So polygenic inheritance is poly meaning many, genic meaning genes. So inheritance and the inheritance of traits due to many genes. Human height. What governs height in humans is actually the additive effects of two or more genes on a single phenotypic characteristic. Human height is governed by multiple genes. Polygenic inheritance is the opposite of pleiotropy, in which a single gene affects several characteristics. Modeling human skin color. Skin color in humans is controlled by at least 3 genes that are inherited separately. For simplicity, we will stick with 3 genes in our example, and these 3 genes are A, B, and C. Each of these genes has 2 alleles, a dark skin allele and another allele. The dark skin allele for each gene, capital A, capital B, capital C, the dominant allele, contributes 1 unit of darkness to the phenotype and is incompletely dominant to the other recessive allele, lowercase a, B, or C. A person who is genotype AABBCC, homozygous dominant for all 3 genes, would be very dark, while a person that has a homozygous recessive genotype for all three genes would be very light. A heterozygote, so a cross of these two in the parent generation, would lead to a heterozygous for A, B, and C, that would have a shade of skin that is intermediate to the first two. Since these alleles have an additive effect on the phenotype, a person who is heterozygous dominant for all three would have the same skin color as any other genotype that has only three dark skin alleles. So I have 1, 2, 3 dark skin alleles here, where I'm homozygous dominant for A, heterozygous dominant for B, and homozygous recessive for C. But here, where I'm heterozygous dominant for all three, I still have 3 dark skin alleles. Okay? And so those two genotypes would result in the same phenotypic skin color. The calculation from a simple model results in this plot of the population according to skin pigmentation. 1 out of 64 having the lightest and darkest pigmentation, and 20 out of 64 having this medium pigmentation. Our simple model illustrated how 3 different genes could produce 7 different skin color phenotypes in humans. However, the humans have many more skin color shades than shown in the model. A more precise model would give a better representation, but not the complete story. Environmental factors such as exposure to the sun, geographical location, and diet strongly influence this trait. Heredity and environment. Many characteristics result from a combination of heredity and environment. A tree's phenotype is genetically determined, but its environment will influence the phenotype as well. Exposure to wind and sun will affect leaf size, shape, and color. Environmental impact on phenotypes are covered in more detail in the last topic in this unit. Epistasis. Epistasis is when one gene, usually in a different locus or chromosome and often sorting independent of another gene, exhibits overriding control over that gene. The albino gene overrides the production of melanin and results in no pigment being produced. Melanin is responsible for the various shades that humans come in. So if a person possesses the albino gene, they're overriding the production of melanin. So we get no pigment, no matter what the genotype would produce normally. This is known as albinism. For humans, regardless of their race or ethnicity, if they possess this gene, they have white skin and hair and light-colored eyes. There are 2 basic alleles for Labrador coat colors. Black is dominant, and brown or chocolate is recessive. There is a 25% possibility of 2 heterozygous black Labradors having a brown puppy. However, there is a yellow epistatic gene which overrides the production of the black-brown phenotype and produces a yellow-coated Labrador. The yellow epistatic gene is recessive, so both yellow genes must be present to cancel the black-brown genes.